everyone. Very good evening. So, Aditya and Shagun, as well as Meghna and Divya. So, it's not Asher sir today taking your session. It's me again. Uh, you've met me already in the last session if you were there. So, good evening, Shagun. Good evening, Aditi. Welcome. So, I welcome all of you once again to another session of NEET Bytes, right? And it's about structural organizations in animals. Very interesting topic, right? And today, me as well, right? You've seen me already. I'm Ghasia. I will be conducting this session today. We'll be taking certain questions. We'll be discussing some concepts, and I hope you have a cool session. You have a learning session. Thank you, Meghna. Welcome. Hi, Saureen. Hello, Sunita. Good evening. Hi, Dimple, Varsha, Namaskar. Aditya, welcome. Right. So, how are you all? How are your preparation moving? And what about your studies? I hope everything is going good. Good evening, Arsalan. Okay. So, let's get started. Let's start the topic today. Let's see how many questions you could get right and what are the things you learn. Very fine. So the topic, as you can see, it's written structural organizations in animals. Okay. I just want to ask a question before I proceed, right? I want to know when you read this term, structural organizations, what do you infer? What comes to your mind? What is the kind of structure you are going to read about? Okay. And what about organization? So based on your journey in biology, okay, like you've started your journey long back and you got an idea about different things in biology, what comes to your mind? Yes, I'm waiting for your responses. What comes to your mind when you see some organization is talking about some structural thing is being made? Okay, so I'm getting some answers from Varsha. She says cells and tissues. Very fine. Aditya says tissues in our body. Okay, very good. So, Dimple, we have earthworm and cockroach in the syllabus. Okay, tissues we talk about. Very good, Aditya. We talk about how to make organs, and organs are coming from tissues. That's good. That's good. So you kind of have an idea as to what we are going to discuss about today. Okay. Now, moving on. So here you can see, right, this particular figure in front of you. It is giving you the glimpse of the very basic understanding that we start, that there are cells. Okay, they are the basic units, the structural units and functional unit. And then when cells who are having similar origin, that they are coming from a common point, okay, and they are performing the similar functions, right? When they group together, they give us an idea of tissue. They form tissues. Okay. Furthermore, since humans, if you talk about, we are complex organisms. So we just do not stop at tissue level. We keep on moving forward. Okay. We talk about group of tissues in that matter. Okay. We talk about group of tissues where we get organ present. Okay, many organs combined together will give us an organ system. For example, digestive system is just one of that. There are many different systems you can recall from your human physiology, if you remember, right? Okay, and then many systems combined together, working continuously for you, okay, giving you a perfect human body, right, that you must be taking care of. You should. Okay, so this was just a basic idea about that. Now, in animal tissues, in this particular topic that we have taken, you're required to know about 4 basic types of tissues that are forming the body, right? Okay, so there are 4 major classes that you can classify them as, okay? Starting with epithelial tissue, right? Connective tissue, muscular tissue, and the nervous tissue. Right below, if you can see in the oval shape, you can find that origin is written. Okay, interestingly, for epithelial, the origin is from all the 3 germ layers, right? It is from all the 3 germ layers. Reason that epithelial tissue is not only covering your body, it is also providing lining to your body. So this is just like as if you are painting your house. Okay, painting in the sense you are doing the whitewash. It's not like you're making flowers. Okay, painting in the sense that you are doing some whitewash. So you do whitewash on the outer face of your house. Okay, you also do whitewash on the inner face as well. I mean, you need to make sure that your walls are covered with some good color and it looks all cool in there. Okay, so it has all the epithelial in your body comes from all the three origin, right? Moving on to connective tissue, it is mesodermal in origin. Okay, muscular tissue being again mesodermal in origin, and nervous tissue being ectodermal in origin. The idea to bring this slide in here that sometimes you do get such easy questions as well. Okay, and remembering them is always going to give you an edge. Okay, now talking about the epithelial tissue, see, when you are going to read about epithelial tissue, follow this flow chart. Okay, divide your studies onto the basis like that, starting with the squamous epithelial, moving on to cuboidal, columnar, ciliated, glandular. You need to know about their structure. Okay, I'm giving you a kind of a skeleton as such. Okay, need to know about their structure and essentially functions, and very importantly, you should know their location. You should have an idea as to where in your body do you find them. This is essential because in our questions also today as well, you will find that there are questions that directly ask you who is present where. So address you will have to remember, okay? Your GPS will not work here. Okay. Moving on to the connective tissue, right? Now, connective tissue comes in all shapes and sizes. I mean, there can be various types of connections if you talk about. Forget about tissues, connections that you have in your daily life. Okay, it's many type of them, isn't it? So some are your ishare friends, some are your chitti friends. We don't have chitti friends any longer, but yeah, we do have different types of connections. The same thing is applied in here as well. Okay, so there's loose connective tissue divided into areolar and adipose tissue. Furthermore, dense connective tissue as regular and irregular. Okay, a specialized connective tissue being skeletal and vascular. Okay, now I'm not going to go into the detail of that due to time constraint, but I really want you guys to follow the flow chart I'm providing in here and divide your study pattern onto this basis. It allows you to remember facts. Okay, now moving on to the third type of tissue, which is muscular. In there you will have to study about the skeletal muscles, those muscles which are kind of your servants. Okay, they follow whatever you want them to do, so that is voluntary actions. Okay, smooth muscles where they don't listen to your will. Okay, they are being guided by the involuntary part, right? And then cardiac muscles which keeps your heart going. Okay, the most hard working people are the cardiac muscles. Okay, so that is how you will be dividing your studies about them. Exactly, these are also involuntary. Perfect. Moving on to the last one, which is the neural tissue. Okay, Jitanshu, basically we are talking about the tissues present in animal system, in human body to be specific, and I am providing you an overview about the different type of tissues you need to study and how would you follow. Okay, what is the schedule that you need to follow. Okay, so in nervous tissues, you'll be talking about neurons that are the major cells, but neurons are not the only cells present. You also need to know about the neuroglia, which are helping hands. Okay, neuroglia are the helping people of neurons. So you cannot neglect somebody helping, isn't it? You need to talk about them as well. Okay, and when you talk about neurons, cell body, dendrites, axon, reflex action, all that you will study. Okay, so I totally understand that this is a vast chapter. Okay, it's a big chapter, but interesting because you get to know about your own system. Okay, you can feel proud of yourself that wow, I'm having neuron, man, and neurons are doing such a sorcery in my body. Appreciate that, totally appreciate it. Okay, very fine. So we'll move into the questions one by one. Okay, we'll try to see the question and then we'll try to understand some of the concepts related to the question. That which concept, if you apply, likewise you will solve the questions. Okay, so shall I begin with the questions, everyone? Are you guys ready for the round of questions? Cool, everyone? Should I start with that? Okay, very good, very fine. So here comes your very first question. Okay. The very first question asks you which cells do not form layer and remains structurally separate. Okay, you have to tell, you have to think and tell the answer that which tissue cells are not forming a kind of a group that you find in other tissues. Okay, these cells are performing their task more individually. They're not present in one over the other, rather they are connected with one end and the next end to the another one. They are connected one by one in a chain manner. They are not present one over the other. This is the difference. Okay, so which cell do you think fits the description? I'm getting answer from Varsha, Meghna, Shagun, Arslan, Dimple. Very good, very good. So you people rock totally. Congo, that's a right thing that you've given. Nerve cells, they do not form a layering pattern, right? They're not present in a bunch. Okay, can you just tell me why? Okay, can you just think from the functional aspect, why are they not present one over the other? And if they are present one over the other, will it be a problem? Do you think so? According to the function, you have to think. Okay, so I'm just giving you a time to explore neurons, right? What do you think? Why they are not forming layers? Why they are one after the other? What suitability is it giving them? Yes, they have to transfer. Okay, they have to take a part in the transmission of nerve impulse that you call as electrical discharge. Can be chemical as well, right? Can be chemical as well. Okay, so they have to gather certain information, and that information they have to travel, they have to make it go further. Okay, exactly, that is why it's not suitable for them. Very good, Shagun, right? They have to pass on the signals. Very fine. So you guys know about them. Very good idea. So I guess this is clearer. This concept is clear to you that it's not cool if we are not having nerve in the layering pattern. It's fine if they are not in layer. Okay. So next question, guys. What do you think? Next question, shall we? Okay, very good, very fine. Moving on to the next question. Okay, here it's a match the following type of question. Okay, you have to find out the correctly paired match. Now here again, I told you, when you are studying about the tissues, the very important thing you have to do, okay, the very important thing you have to do, you need to remember their address. Okay, you need to remember their address. What is the meaning? It means that you need to know which one is present where. Okay, which one is present where. This thing you need to remember, because you get straight questions, right? This is such a straight question, right? Which one is present where? Location is very specific, so please make sure you do that. So I'm getting many answers. Okay, let's try to understand this question. Okay, inner lining of salivary ducts, it doesn't have ciliated epithelium. Okay, ciliated epithelium is not of a task in the ducts. Okay, so that is not a correct match, right? Moist surfaces of buccal cavity, again, it's not glandular, right? So you are guessing it very correctly. Tubular part of nephrons have cuboidal epithelium. And so should you know that cuboidal epithelium, it's simple epithelium. It has cube-like cells, okay? And just to tell you that you find cuboidal epithelium at two places, okay? You find this particular epithelium at the ducts of glands, very important, keep this in mind, okay? And you also find this in the tubular region of nephrons, okay? In the tubular parts of nephrons, you find this particular epithelium. Exactly. So for secretion and absorption, I was just coming on the function, so you told it already. Very good. So as we got to know from Aditya, that their function is to absorb and to secrete, and that is why they have a large size. Okay, very good. So moving on to the next question. Okay. Hello, Parimala, welcome. Okay. In which one of the following preparations are you likely to come across cell junctions most frequently? Okay, now here what happens in this type of question, again, see, these questions are easy to solve when you have thoroughly read the tissues, you've thoroughly read their structures, and you've thoroughly read their functions. Okay, because everybody is showing a distinct feature. Okay, it's not common for all of them. So when you are going to read about ciliated epithelium, you will find that ciliated epithelium is present in the cell junctions. Okay, so that's right. That's correct. A guess I've gotten from Varsha. It's ciliated epithelium. Now my question to you is why? Okay, no problem, Gaurav. The question is, why do we have ciliated epithelium at the cell junction? Before answering this, you need to tell me the function. If anything you know about the function of ciliated epithelium, right? Anything you know about ciliated epithelium? What does it do, basically? What is the function of ciliated epithelium? Yeah, that's what I'm asking. I'm going to answer this. I just want to have few words from you guys. So what is ciliated epithelium doing? Yeah, I'm explaining. Protection. Okay, apart from protection, Ayush, apart from protection, anyone? Passage in one direction. Okay, since it has gotten cilia present over it, okay, let's say this is a cell, right? And it has gotten some projections present over it. Now, what is it doing? The cilia is allowing, the presence of cilia is allowing the cells to act like, okay, like traffic police. Okay, they can guide, right? They can guide the molecules, right? They can guide the substances, guide the molecules, and allow them to move from one place to the another place. Exactly, they're helping in moving. And in the cell junctions, so why do you have cell junctions? Are basically in order to pass on things from one cell to next cell. That is for passing on. Okay, and this is the reason you get ciliated epithelium there. You want somebody actively doing the task. Okay, so that is not for locomotion in this case, Ishita. Okay, in this case, your ciliated epithelium is not taking a journey. Okay, I'm in head, and next time I will go to heart. It's not like that. It's present at one place, but in one place also it is doing the pushing forward. Very good. Okay, but nice name you've gotten, Devdas. That's a right answer. So I guess you've understood this. So ciliated epithelium is somebody who is performing its task of cell junction. Okay, you want somebody to actively push out something. Okay, very good. Next question. All right, this is about the muscles. Okay, this is about the muscles. Now, you all must know, before I discuss this question, we must have an idea that in muscles you find something called H zone, okay, something called A zone. Am I right about that? This is what you find in the muscles. Okay, right? And H zone is characterized by the presence of thick filaments that are also termed as myosin. Okay, also termed as myosin, right? So they are asking you, how come you have H zone present? Okay, if you read the options one by one, you will arrive at an option which is telling you that H zone is basically central gap. Okay, it's a central gap between two actin filaments. So A zone, that is a dark band, that is characterized by dark band, okay, and H zone is light band. And if you have a little bit of idea about the picture of muscle, you'll find that between the A zones, the dark band, okay, you have the H zone present. Okay, this is the H zone, the light band, right? This is some sort of a, like, if you are need to have photographic memory, you can click the picture and keep it safe in your mind. It comes back. Okay, so C is the correct answer. Okay, the central gap between actin filaments extending through the myosin filaments in the A band will be giving you the H zone. Okay, it's present between the A bands. Okay, so this is the answer here. All right. Okay, so moving on to the next question. This is about connective tissue, basically the loose connective tissue, okay, which is your areolar connective tissue. Simply asking you this question, simply asking what is the function, okay, because it is connective, it has to do joining. This is understandable. If it is connective tissue, it is joining something, right, per se. What is it joining? What is the function it is performing? Okay, so fat body with muscles, integuments with muscles, bones with muscles, bones with bones. What do you guys suggest? So I'm already starting to get answer from Varsha. I get one. So I just want few more people to provide me some answers. Okay, okay. All sort of answers are coming. Very good. I wish if it could go beyond A, B, C, D, E, F, G, H. Okay, so B is the right answer, right? In this case, B is the right answer. Basically, understand what happens. Your areolar connective tissue, okay, areolar connective tissue, it is richly supplied with nerve cells and blood vessels. It has a rich supply of blood cells, nerve cells, sorry, and blood vessels. So what does it do? It is connecting, okay, it is connecting the skin epithelium, which is integument for that matter, okay. It's connecting skin epithelium with the muscles down below it, okay, with the nerves and with the blood vessels. This is the function in actuality, okay. And who does this function? None other than areolar connective tissue, okay, none other than areolar connective tissue, okay. Understandable, right? So you've gotten your answer, Gaurav. Very good. Yes, present beneath the skin. That's right. That's right. Fine. Understandable, guys, I guess. Okay, then I will move on to the next question. Here it is an assertion and reason question, an easy one. Okay, this is something that you remember. Okay, now, assertion is that cartilage and bone are flexible and rigid connective tissue respectively. Okay, they are saying that cartilage is flexible, bone is rigid connective tissue. Do you guys agree to this thing? May I have your agreement for this assertion? May I have the agreement for this assertion, or maybe disagreement for that matter? Maybe you can disagree. That's not a problem. So let's see what is how many people go with agreement. So is it, tell me if the assertion is right. You tell me if the assertion is right. Okay. So Varsha is agreeing. Shagun is also agreeing. Dimple agrees. Maria agrees. Sorin agrees. Wow, so much agreement I have gotten. Very good. So that's true. Okay. Assertion is true. Right. Now let's go to the reason for assertion. Reason says that blood is a connective tissue. Okay. Now reason is right. Okay. Reason is right, but reason is not explaining assertion. Reason is not explaining assertion. Both the statements are right in themselves, but reason is not the explanation of assertion. You understand? Blood is a connective tissue. Other cartilage and bone are also flexible and rigid, but how are they both? How is the reason substantiating for the assertion? Okay, so we arrive at option number B, right? Both assertion and reasons are true, but reason is not the correct explanation of assertion. Right? Such a fun question it was, isn't it? So this is one of the types of pattern that you get when you are giving for the AIMS examination. Okay, very good. So I guess this is understandable to you guys. Okay, it's understandable. Right. Moving on to the next question now. Fine. Okay. The question is asking you about the gap junctions. Okay, it is telling you, asking you the function of gap junctions. Before I move on to gap junctions, do you guys remember somebody named as plasmodesmata? Do you guys remember somebody called plasmodesmata? Any remembrance of this person? Do you remember that? And if you remember, where was it? Where did you meet this guy? That's right, that's right. I'm getting right answers from Varsha, Aditya, Meghna, Shagun, and Arsalan, Mohit Raj. Sorry, in that school. Okay, right. So answer you have given. I just wanted to know, where have you seen plasmodesmata for that matter? Just connecting between cells. Where? Which cells? Plants. Perfect. Okay, Meghna and Gaurav. Yes, Devdas as well. Yes, okay, very good. Now you found it in the plants. So basically, the gap junctions and plasmodesmata are having the similar functions but in different organizations. Okay, plasmodesmata was something that you came across in the plants. Gap junctions are something that you come across in animals, however, performing the same function. Okay, similar function, I would say. right? So on the basis of that, we can choose option number C, suggesting that gap junctions are basically facilitating communication between adjoining cells, two cells present right after the other in adjacent fashion, by connecting the cytoplasm for rapid transfer of ions, small molecules, and some large molecules. So that is just like a channel you have. That is a passage, a channel where people can, move from one cell to the another cell. Okay, this is the idea. So keep in mind, with the gap junction and the plasmodesmata, you do not confuse. They are present in different organizations. That is, in plant kingdom you find it, in animal cell you find it. Okay, not plant kingdom, I should say. That is more in the plant cell, come across in angiosperms, right? Next question now. The type of epithelial cells which line the inner surface of fallopian tube, bronchioles, small bronchi are known as. Okay, see, in this particular question, you must have an idea what is happening in fallopian tube, what is happening in bronchioles, and what is happening in a small bronchi. You need to have a little bit idea about their function. So what happens in fallopian tube, if you remember, fallopian tubes are collecting ovum from the ovary. Okay, they're collecting the after the ovulation, they are responsible for collection. For that matter, you need some finger like structure. Yes, Aradhya, all need cilia. Bronchioles and bronchi are pushing the air you have taken in forward to the alveoli, right? All of their functions is to do some pushing and pulling task, isn't it? That is the common functionality, right? And that is the reason all of them will have same type of epithelium, that happens to be ciliated epithelium. Okay, that happens to be ciliated epithelium, because cilia, as I have already told you, it's responsible for certain pushing and pulling task. Okay, but cilia is not allowing it to move, it's not moving in there, but yes, allowing it to do certain task of pushing and moving things. Okay, that is why we say that ciliated epithelium will be there, and that's true. Okay, that's true. So you understand that how we arise at this particular answer, because of the function we know, that happens in fallopian tube, that happens in bronchioles, that happens in small bronchi. Okay, guys, understandable this one is? Okay, now moving on to the next question, that is about your friend. Okay, if it is farmer's friend, then it is our friend as well. It's interconnection, food chain works here. So if farmers are growing the food, then we are eating it. If somebody is their friend, it's our friend as well. Okay, and that's about earthworm, the ferritima as you call it. Okay, so they are asking you a very special feature that you find in earthworm. Okay, something special. This question came in your aims. Okay, this is an aims question, right? And very easy to guess. Fine. So I'm giving you some time. Come across. What is something that you think is special in here? Fertilization occurring inside the body. Typhlosol is present, which is providing effective absorption area in the intestine. S-shaped setae are there. Long dorsal tubular heart is there. What is the right answer? So I'm getting one from Maria Rizvi, Aradhya. Okay. Right. S.K. Hussain, Varsha, Shreya, Gaurav Gupta. Perfect. So correct answer is B. Okay. Now you guys know that the intestine of earthworm bears the pocket of enfoldings. Okay. Pockets of enfoldings. Right. And that this is similar to the villi that you have in your small intestine. Okay. Similar to the villi you have in the small intestine, which is doing similar task, absorption, providing greater surface area. Okay, the same thing it is doing there. Okay, so typhlosol is the right answer. And where exactly do you find typhlosol? Okay, so typhlosol will start from 26th segment. Okay, and it will go till end. Okay, it will go to the end segment, but you do not find it at 24 and 25 segment. Here you will not have the typhlosol. Okay, start from the 26th segment. All right, 26th segment. Very good, very fine. So this was one of the very special features earthworm is having. Okay, now moving on to the next question. Here again, you have to find the correct match in about the things present in earthworm back again, right? What is their function? Is it rightly matched or not? Okay, see, first one is telling you that clitellum is secreting cocoon. Okay, if you've any idea about the earthworm's body, you would simply say that, ma'am, this is true. Cocoon does secrete the clitellum does secrete the cocoon when the reproduction is to happen. Okay, right? So this is the right answer. Gizzard absorbs digested food is false. That doesn't happen. It is basically for grinding the food. Okay, it is basically for the grinding purpose. It is grinding the food. Okay. Setae are for defense. This is also false. Setae are for movement. Okay, they are for movement. And tiflosol we have already seen. Is it not? It's not storing anything. It is for the absorption. It facilitates absorption of the food in intestine, right? So the only answer we left is with the A. Yes, exactly. You're right in that, Varsha and Aradhya. That's locomotive. Setae are locomotive. So this is the right answer. Fine. This was very easy. You just need to know different structures of earthworm and you can simply have the right one. This also came in AIMS. This is also an AIMS question, right? Okay. Moving on to the next question now. This is about your friend. Okay. This may not be farmer's friend, but definitely ours friend, you can say. Periplaneta americana, the mighty cockroach. Okay. The mighty cockroach, we are talking about it. Okay, so which one is correct statement regarding the Periplaneta americana? Cockroaches, perfectly. Okay, so if you read all these things, okay, for that I really want you people to have a thorough study of cockroach. No matter how much you hate it, it's okay, but you have to study about it. Okay, sometimes it happens. It is good to know somebody who you hate, isn't it? So you know where to apply the uh, task perfectly. Okay, so I'm coming with very right answers. I'm getting the right answers. Congratulations to Aradhya, Shagun, Meghna, Mohit, Suri. Very fine. Males bear a pair of short thread-like anal styles. Now, you should know that you can distinguish between females and males in case of cockroaches. That is distinguishable, right? So in males, you find they do have anal styles present, and anal styles are something like thread. Okay, they are thread-like structures, unsegmented, unjointed. Okay, so this is a very peculiar feature you see in male. Now, next time when you come across a cockroach, instead of killing it, have some patience. Go at the anal styles. Try to find out whether you are killing a male or you're killing a female. Okay, and then you can proudly boast about yourself. I have killed a male today, or I have killed a female today, right? Because you know what is a female, which one is a female and which one is a male. Okay, right? Earthworm is there and as well as cockroach. Frog is not, Aradhya. Okay, now moving on. What external changes are visible after the last molt of cockroach nymph? Now, this is also a last question. Okay, and in the last question, we are talking about the last molt of cockroach nymph. Now, before I tell you the answer, before you tell me the answer for that matter, because I know you guys know it, you should know that cockroaches are having something called as porometabolous type of development. Okay, keep this in mind. And what happens in this development, it occurs through nymphal stages. Okay, it occurs through nymph stages. Okay, and there are 13 molds that has to happen before you get a proper cockroach. Now, how can you just kill a cockroach like that? Can't you see how much hard work it is putting into it? 13 molds are happening, 13 times molting is happening, and then a cockroach is coming out. Okay, several times in includes 13 times for that matter. You need to have some sort of sahanubhuti. Okay, that sort of because you know, you can say, cockroach, I know about you. You've done so much hard work to become a cockroach. So what external changes will you find after the last mold, after the 13th mold? The only difference that you will find is that both forewings and hindwings will come out. Otherwise, nymph looks exactly like a cockroach. Okay, it looks like cockroach. the adult cockroach, the young cockroach. The only difference is that nymph doesn't have the proper wings, hind wings and forewings. Okay, what it has is the wing pads. Okay, it has the wing pads, but once it becomes a cockroach, it will have the wings. So this is the only thing that is going to develop in the last molt. Okay, so this is the correct statement, which is option number B. Okay, now this was the last question regarding the last molt as well. I guess you have gotten some idea as to how will you be studying the tissues, okay, and how will you be applying it in the questions, how will you be testing what you've learned, right? So with that, I wish you a very happy learning. Okay, keep the flowchart in mind that I have given in the starting of the video. Okay, if you follow that, it's not like you have to follow that, but if you do, it's somewhere easy, it somewhere allows you to have a easy pattern of understanding. Okay, so with that, people, I take your leave now. We will meet again in the next session, okay, with some more interesting questions, okay, and with some more interesting examples, right? And thank you for being here, for actively answering and talking about earthworm and cockroach. That was real fun. So I take your leave. Stay in good health. Enjoy your time. Bye bye.